during development is as essential for an understanding of evolution as are the study of mutation and that of selection. I kind of want to change this, yeah, to say that a study of the regulation of genes during development is as essential, if not more essential, for understanding evolution and for understanding development. And so we'll go into this towards the end of this lecture. So up until so last week's lecture, I had introduced this general framework for thinking about patterning. Essentially, we have gradients or signaling interactions that set up patterns. Here we're looking at the blastoderm or the neural tube. All of these are examples of gradients of signaling proteins that will set up a particular pattern. Regulatory elements that will then control target genes. And so these signaling factors are switching on particular factors downstream that in turn are turning on the expression of target genes. And so these transcriptional networks that are initiated by these signaling molecules are the ones that actually generate the pattern. And so these networks have various different components of feedback, inhibition, and repression that strengthen and formulate particular patterns in different parts of the embryo. So I think this this kind of way of looking at how patterning happens is quite is attractive. It's probably correct as well, and there are probably nuances that you'll pick up when you go further into the details of all of these transcriptional networks. What I want to talk about is these downstream components. So these genes that are now regulated and how they're regulated and what does that mean? And so we know that particular transcription factors, for example, enact developmental programs. And here you're looking at some work, some quite old work actually, from about 20-25 years ago, that looked at the function of a gene called OTX. OTX is the vertebrate homologue of a Drosophila gene called orthodentical. And so what you're looking at here are mouse embryos, and so this is a embryonic stage. It looks like about E12.5, and this is a newborn pup. And what you can see in the wild type is a very nice embryo with a forebrain, midbrain, hindbrain. Here you can see the pup, and in the mutant, the structures anterior to the forebrain are lost. And so this is very clear here in this embryo, and even clearer in this postnatal embryo. Various reductions in the gene dosage cause various reductions in the head phenotype as well. But essentially, it seems as though OTX is important for enacting a developmental program that leads to the formation of the anterior neural ectoderm of the embryo. Okay, so when we want to understand what the control elements are for OTX2, what we can do is look for the enhancers that control its expression. And so if you have a question about what enhancers are and how they're distinguished from promoters, hold on for a little bit and we'll talk about that in a few slides' time. But here, what you can see is one strategy that this group of Shinichi Aizawa employed to look for the control elements that control OTX2 expression. And so what you're looking at here from G to K is the endogenous expression of OTX2. G to K, yeah. And these slides here from A, B, A, B, C, D, E, F, and L, what you're looking at are the activities of various upstream components of OTX2. And so what this group have done is taken a 200 kb fragment of the OTX2 gene and split it up into different pieces, put these pieces onto a reporter gene, and then made a transgenic mouse to ask which expression, what domain of expression these reporter genes drive. And what you're looking at here is this number 12 fragment is showing expression that kind of looks like OTX2. This number 4 fragment shows another component of the OTX2 expression as well. And so in this paper here, what this group suggested was that this fragment 12 was important for driving the anterior expression of OTX2. And so if you want to then understand whether this really is important for everything that OTX2 does in these stages, what you want to do is then remove this, the enhancer sequence from this fragment. And so I should tell you what enhancers are. So I think everyone knows enhancers and how they're distinguished from promoters. Is that correct? Does anyone not know?
I don't.
Okay, thanks. So what enhancers are generally elements that are generally in cis, so in the same genomic fragment as a gene of interest. So this is gene A. They're upstream of the promoter or in different places from the promoter. They act to enhance the ability of a promoter to promote transcription of the gene, essentially. So this distinction between enhancers and promoters is important, but it's not so important. It's now thought, and if you talk to Dimple, for example, it's now thought that enhancers have a kind of basal promoter activity, and promoters themselves can have basal enhancer activities as well. But essentially, for this lecture, we're going to distinguish. We're going to primarily talk about enhancers as separate from promoters. So promoters are driving a basal level of transcription of our gene of interest, and enhancers, of course, are then enhancing that transcription. We have also, we can consider as well silencers, and so silencers will stop expression, basal expression, from a particular promoter. And we can also have insulators, which prevent a cis-regulatory element of here gene A influencing gene B. And without an insulator, for example, you start getting ectopic gene activity of different genes. Okay, so these are enhancers. We're trying to think about how we can control the expression, how we can identify whether this enhancer here, this enhancer 12, or the enhancers contained in this fragment 12 are the ones that are important for the function of OTX2. And so,
Raj,
yep.
Can I interrupt you for a moment?
Yes.
Yeah. So in the previous experiment, so if I followed you correctly, they're exogenously expressing different enhancer sequences and assessing its impact on OTX2 functioning, right?
No. Okay. So what they're doing, they're not looking at OTX2 function. So that's what they do in the next slide. What they're doing is to see if these enhancer sequences can drive, can recapitulate the expression of OTX2. And so what you're looking at here, these KI labeled slides, is a reporter for normal OTX2 expression. And these slides, these panels here, number 12 and number 4, are showing the what expression is driven by this fragment 12 and this fragment 4. And so the way they've done it is, and I should have put a slide for this, is to take the fragment, hook it up to a reporter gene, and in this case it's beta-galactosidase, and then injected it into a mouse embryo, and then just asked what expression was being driven. And so this is a very, very simple transgenic mouse experiment. Okay.
So is it like monitoring the spatial, control of gene expression as mediated by these various enhancers?
Yeah, absolutely. Yeah, that's the way to look at it. And not just spatial. I mean, here we're looking at static images, but you do get a feel for temporal expression as well. Okay.
Yeah.
Okay.
Thanks. Yeah.
Great.
Raj, how are they? How do they?
Sorry, Shubha. Can you talk again? I think I lost you.
Enhancer in each of the cases.
Sorry, can you repeat your question, Shubha? I for some reason I missed what you were saying.
How do they know that? How do they activate or ensure that one particular enhancer is activated in each of these cases?
Okay, yeah, that's a so it's a good question. It is a good question. Sorry, I shouldn't hesitate when I said it's a good question. What I wanted to say is that all they know is that this fragment here is driving expression of this reporter gene here. Okay, and so of course this fragment 12, could contain multiple enhancers. And the way they would find that out is by then taking various fragments of this 12 and putting it upstream of a reporter gene. So this reporter gene is not normally expressed in mouse embryos. It's a gene that, when expressed will give a colorimetric reaction in the presence of particular chemicals to make a blue precipitate. And this gene is a bacterial gene, beta-galactosidase. It's not, like I said, it's not expressed in the mouse normally. And so here, this is kind of now newly introduced expression that's being driven from just this fragment here. Okay, is that kind of understandable?
So you mean to say it depends on the particular region of the developing embryo? Yeah.
So hold on to those thoughts, Shubha, because I'll explain this in the context of another experiment in a few slides time. Okay.
Okay.
Right. So would it depend on the presence of other factors which would be binding on the enhancer at a particular position? That's why they are expressing at these positions.
Fantastic. That's probably where we're driving to. Again, hold that thought, yeah, and we'll go into this in more detail in a few slides. Okay. And then again, I'll ask your question, and then we'll use that as a way of kind of discussing all of this, okay, and bringing everything together. Okay. Nice. So what they've suggested here is that this fragment 12 contains the enhancer that drives anterior expression of OTX2. Okay. So, like I said, this is the normal expression of OTX2, and this is what this enhancer 12 is driving. And so to check whether indeed enhancer 12 does have the sequences is purely responsible for OTX2 expression, what they've done is to knock out the enhancer. So they haven't touched the gene, they've removed the enhancer. And this is what you're seeing in this slide, in this panel down here. And what you're seeing is, so here this is the normal embryo, and they're looking at various regional markers of the brain, 6-3, FGF8, GBX2, for example. And then they're looking at an embryo where the enhancer that they've this number 12 enhancer that they've characterized is deleted. And what they find is that there are subtle defects, not so subtle, they're quite profound defects in the anterior patterning of the mouse embryo. But it's not the same as removing OTX2 function itself. And so what they've concluded is that this enhancer that they've identified does indeed control expression and control regulated localized expression of OTX2 and contributes to the overall function of OTX2, but not completely. They're missing another enhancer that's probably found in some of these other sequences that acts maybe redundantly with this enhancer to drive OTX2 expression. Okay. So the idea of, sorry, I'll come back to this line of thought. So another way that enhancers can be discovered is by comparative genomics. And so I want to very rapidly talk about this paper here. It was published 8 years ago and from the group of Kiyoshi Kawakami in northern Japan. First author is Shigeru Sato. And so what Shigeru did was to look at 6-1 expression. And so 6-1 is a transcription factor. It's involved in various processes during development, and its expression is generally conserved. And so they know that. So it's known that 6-1, for example, is found in what will become the ear. It's found in what these structures here called the dorsal root ganglion. It's found in the notochord, and it's found in the olfactory epithelium across vertebrate development. And so if you know that the expression of a gene is conserved throughout animals, this gives you a way of then looking for enhancers that may control this expression. So before I go on, does anyone want to hazard a guess of how they've done this?
Well, they can actually compare the sequences of different organisms, and maybe they can compare it with ones whose enhancer sequences have already been determined. And so if there is a similarity in the sequence between the two, we can get a rough idea about the sequences which might function as enhancers also in the other organism. Yeah,
that's very good. That's exactly what they did. And so here's a normal expression of 6-1 in chick, and so you can see this beautiful expression. It's expressed early in the neural tube. It's expressed later in these kind of segments, the somites. It's expressed here in the dorsal root ganglion. You can see it expressed in various parts of the embryo. And so, Nivedita, you're right. That's exactly what they did. They compared. So they knew that the expression of 6-1 was essentially conserved all the way to fish. And so they lined up the sequences around 6-1 from human and compared it to mouse. Okay, so here the base genome is mouse, and what you're looking at in these peaks are sequence similarity. And of course, when we're thinking about where different animals lie in relationship to the mouse, humans are quite close to mice, and so there's much more similarity outside of the gene. A possum is, it's a mammal, and so it's kind of similar. Chicken, we're getting a little bit further away evolutionarily. Xenopus as well. And so in this way, in this kind of comparative way, you start honing in on particular highly conserved, ultra conserved sequences that are outside of the coding sequence of 6-1. And so let's zoom in here onto the 6-1 sequence. And so here you can see the coding sequence, so the introns and exons of 6-1. And what they're looking at is about 300 megabases. So they're looking at about so here 240 megabases upstream and about 100 odd megabases downstream to look for sequence conservation. And in the course of kind of defining these highly conserved sequences, they've narrowed in on these numbered sequences here. Okay, and so they've classified all of these elements as being highly conserved, all the way down to zebrafish, for example. Here, this fragment here is not found in Teleos, but is found in stickleback, for example, Medaka, and so on. Okay, and so they took the same strategy as the OTX2 group, Aizawa's group, and what they've done is to take out these sequences, clone them upstream of a reporter gene, and in this case it's beta-galactosidase, and make transgenic mice and ask where expression is being driven. And so what they find is very beautiful. This is fragment 8, or piece 8, which is this highly conserved sequence here. And what you can see is expression being driven in the dorsal root ganglion. So these are going to form sensory neurons of the trunk, and in these sensory neurons of the head as well. Okay, this sequence 12 here is showing very beautiful expression in the endoderm and in the notochord and roof plate. Sequence 21 here, which is this highly conserved sequence that's found in the control elements all the way down to zebrafish, is showing expression in the nose and in the ear, and so on. And so this gives us an idea that perhaps these enhancers are kind of modular, that they act almost independently to control different aspects of the expression of a particular gene. Okay.
Raj?
Yeah.
Yeah, so as you said, after we identify these evolutionary conserved sequences, these are cloned in mice and along with reporter genes, just like in the previous experiment, right?
Yeah. Yeah.
So in the actual previous experiment that you described, how I was wondering how they selected the specific fragments which they were testing to have or not have the enhancer sequences. And so did they use a similar comparative genomics analysis?
No, they didn't. And so in the previous analysis, they just did what's called a shotgun approach. So rather than honing in on these particular highly conserved sequences, they just took fragments of this genomic sequence and cloned it in front of a reporter gene. And that way, then, if one of those fragments showed expression, then they took that fragment and cloned it down even further to narrow in on the enhancer. So they're kind of presupposing that there may be no conservation, even though there turns out to be quite significant conservation in the upstream and downstream sequences of OTX2. But here, Sato knew that 6.1 expression was highly conserved, and so he took this approach, which was to do this comparative genomics approach. The comparative genomics approach is something that was introduced quite a long time ago, and so this was something. I mean, Sidney Brenner was a big proponent of this approach, and Brenner had used Fugu as a way of doing comparative genomics. I don't know if anyone knows why you would use Fugu, which is a pufferfish, of course. Why would you use Fugu over any other fish to do comparative genomics? Anyone? No? Okay. So Fugu has the smallest vertebrate genome. So it seems to have been stripped of any kind of what people have called junk DNA. And so any comparative approach done using Fugu is generally fairly rigorous. Okay. Okay, so let's start thinking about what is an enhancer. So I've kind of explained what an enhancer does, yeah, but we still haven't really understood what's actually inside an enhancer. So kind of what does an enhancer physically consist of?
DNA sequence.
Yeah. So it's DNA, it's sequences. So sequences that do what?
It can bind to some proteins.
Yeah. Okay.
It would be a
And-
region where there would be multiple transcription factor like things binding. So it will have like binding motifs for many transcription factors.
Okay, that's good. Yeah.
Yeah.
Yeah. Okay. So that's exactly what it is. So an enhancer is essentially, you can think of it as clusters of transcription factor binding sites. There are different ways of thinking about these clusters, and my feeling is that both are fairly accurate, and it depends on the context. And so, for example, these motifs, these clusters all have a particular logic, which this review here, which is a fantastic review, and I kind of highly recommend you read it, even if you're not interested in developmental biology, if you're interested generally in the idea of gene regulation in different contexts, this is really, really nice, this review. So you can get motifs that kind of these clusters with a particular logic, which is called a grammar. And so here these clusters act cooperatively to direct DNA binding. And so here they need to be in a particular orientation, in a particular spacing to allow, number one, the transcription to be enhanced. But more importantly, remember, a lot of these enhancers are going to be shielded by nucleosomes as well. And so maybe many of these enhancers are under compacted chromosomes. And so you're going to need to get at them as well. And so enhancers will need to, or the factors that will bind to enhancers will need to somehow evict nucleosomes so that they can bind. Okay, so you can get, of course, this enhancersome model, what they've called a billboard model as well. And so here, the cooperativity is indirect. It could be additive as well, and all of these individual transcription factors will bind to the sequences within the enhancer independently of each other. But somehow they will promote gene expression in a different place. And of course, you have mixed modes where you can have some of the motifs within an enhancer showing cooperative binding, and others showing this non-cooperative indirect DNA binding as well. So all of this is to say that when we're thinking about the actual mechanics of the enhancers, how transcription factors will bind to individual motifs, it's good not to have a fixed idea about how this would happen. It's very easy to think about this in terms of kind of enzyme kinetics or whatever, where as soon as you have one transcription factor binding, it promotes the binding of another one, and so on. That could be true, that may not be true. And so those kind of experiments just need to be done. As I mentioned, the important thing as well is kind of the eviction of nucleosomes. So opening up the chromatin so that it can access particular transcription factors. So there are a group of transcription factors called pioneer factors, which whose job it is to prime an enhancer for gene activation. It may not even prime directly the enhancer. They may bind to the locality of the enhancer and cause changes in the chromatin structure to enable then other transcription factors to bind later. Okay, good. So as well as and so when we're thinking about the mechanics of how an enhancer would work, once transcription factors are bound, of course, you'll probably get coactivator complexes as well that then stabilize RNA polymerase or other kind of basal transcription factors that are necessary for gene transcription. Okay. The important, so what this kind of, go back, I'm going the wrong way. What this kind of modular concept of enhancers then gives you is kind of a huge amount of flexibility. And so we know that genomes will duplicate. And so maybe some 50 years ago, Susumu Ono brought in this kind of articulated the expression of genome duplication as a way of evolving new functions for particular genes. He suggested that birds and mammals had duplicated their genomes, and these duplicated genomes could then take on new functions. So let's think of, for example, this gene X here. It has two enhancers that drive expression in the face and the limb. If you have genome duplication, you can sub-functionalize this gene X. So you get, for example, X.1 and X.2 driving expression in just the face. So the limb enhancer in X.1 has been lost. And when I mean lost, it's just by the process of random mutations. This enhancer has not been deemed necessary and has not been selected for in gene X.1. That's because X.2, which is now taking over the function of X in the limb bud, and so here the face enhancer has been mutated. Here another concept is of distributed gene dosage, where now these enhancers are less strong, but because there are now two genes, they both need to be expressed in both the face and the limb. And here is probably one of the nicest ways to think about it, is to which is kind of neo-functionalization, which is giving new functions to a particular gene, because now X.1 gene X is duplicated to X.1 and X.2. Maybe X.2's face enhancer has gained a mutation which now drives expression in the midbrain, allowing now gene X to be functional in the midbrain and allowing maybe new activities and new functions to be ascribed now to gene X just in the midbrain. Of course, we can, there can be kind of some spontaneous emergence from ancestral DNA sequences. So gene X could just by genetic drift, could start, the appearance of transcription factor binding sites could emerge, and these could then start driving expression in particular regions of the embryo which are necessary and enable a new function to be explored by gene X. Similarly, the co-option of an endogenous retrovirus could drive new expression in different parts of the embryo, and there's some evidence that endogenous retroviruses are important for driving expression or for gaining for some genes gaining the ability to be expressed in various parts of the brain, for example. And of course, the remodeling of enhancer promoter communication could also happen by flips and chromosome translocations. And so in this way, you're getting the modules of the enhancers are kind of being repurposed just by simple evolutionary processes. And this is allowing the gene that it's driving to then gain new functions in new places. Okay.
Raj?
Yeah.
Yeah, I cannot really understand what purpose subfunctionalization serves, because even without subfunctionalization, it seems as if the expression of the gene remains the same. Just in this second case, we have two different physically separate regions of the genome governing the expression.
Yeah. Yeah. So maybe it's worth thinking about subfunctionalization in the context of neofunctionalization, this repurposing existing enhancers. And so if you now have two genes that, and you're right, they're serving the same purpose. But it could be that, the sequence now, the coding sequence of gene X is optimized for whatever the function in the limb is, number one. Number two, this is probably more attractive, and this probably is more closer to what's actually happening. Here, if you now duplicate the whole genome with the face and the limb enhancer, if instead of this X here, you're getting this enhancer being repurposed, so that now gene X.2 is expressed in the midbrain as well as the limb. This is probably what's going on. Okay, so you're right. Functionalization may not, from just this diagram, doesn't appear to play a particular role. I mean, it's evolution. It's all kind of random anyway. But you can see how, this fixation could occur and allows this limb gene to explore different ways of regulation and different phenotypes without having to worry about a kind of a consequence on whatever the function in the face is. Okay.
Yes, I get it.
Yeah. Okay. So yeah, this kind of idea that these modular enhancers could be used to evolve new functions and new expression domains is kind of a way of thinking about enhancers in development as a way of just starting to generate novelty as new species evolve. And this is something that Sean Carroll, for example, if you watch any of his lectures, he makes a big deal about thinking about these modular enhancers evolving new functions and new expression domains. And a lot of this work has been done on cichlids, which is a fish living in lakes in Africa, and in stickleback as well. And so if you're interested in this kind of thing, I recommend you start thinking about cichlids and sticklebacks. So let's go back to the actual mechanics of enhancers. And so I think, as you all know, enhancers also influence the higher order structures to regulate developmental gene expression. And so what we're thinking about here are what are called tags, which are I think they're topologically associated domains, something like that. And so TADs are kind of ways that enhancers will cause the chromatin to loop in particular ways, so that now we have new interactions between different, very long-range sequences to control gene expression of a particular gene. So what you're looking at here is kind of a interaction map across some hypothetical sequence in mammals or in Drosophila. Here is 1 megabase in mammals, and all of these dark red squares show where you get interactions between those two sequences. And so remember that this is a line. It's all in cis, but somehow this sequence here is interacting with this sequence here every now and then. Every now and then this sequence here and this sequence here is interacting, and so on. And what this suggests is that you're getting looping of the chromatin to form new interactions and new partnerships of different transcription factors that may be found over here, but because the DNA is looping, they'll come together and generate different means of gene regulation. And so, of course, I'm not doing this justice. I think Dimple will lecture about this next semester, and so you can quiz her about it as well. Okay. Good. Any questions here? Few.
I had a question.
Yeah. Sure. Sorry, I think is it my connection that's terrible?
Nope.
Couldn't hear her either.
Okay. And Adita, I didn't hear you. I didn't catch your question. Can you try and ask it again? Yeah, I can hear you.
So what are the cues that actually initiate the TAD domains to can you hear me now?
I heard you till I heard you till 2. Is it to loop? Sorry, Anadita, I didn't hear you. Okay. So,
I have a quick question.
Yeah.
So when we talk about these TADs, like the enhance, so these are usually the enhancer promoter or like enhancer or any, I mean, gene any of the sequence interactions happening more frequently as compared to where with the other sequences nearby.
Yeah.
So how would they look in different cell types? Because so in the developmental scenario, when we talk about like these promoters being regulated by these enhancers, and these enhancers are different in different cell types, like regulating these promoter genic expression. So would the TADs also look different in these different cell types?
I guess so. Yeah. You know, I would think that they would look really different, and there'd be a certain kind of you know, dynamic looping depending on depending on the cell type, maybe depending on time as well. I think the technology has just got to the point where we can start exploring this, but it hasn't been there up until now. Okay, so you're right. This can be looked at now. I mean, where it had been investigated is in polytene chromosomes, and so I guess probably many of you know about these highly hyper expanded brush border chromosomes that are found in Drosophila salivary glands. Yeah, and so that has given some ideas that these TADs would be specific to a particular cell type, but beyond that, I'm not entirely sure what's been investigated since then. Okay.
Sir?
Yeah.
An answer can bind to multiple transcription factors at the same time.
Yes. Yeah.
Sir, but how is that possible? Because the enhancer has to take some structure in order to regulate gene expression.
Mm-hmm.
So if one transcription factor is having a particular expression of the gene, so multiple transcription factor are binding. So an answer can take a structure one at a time, na?
So it may not, yeah. And so I think this goes back to this kind of model here. Okay, yeah. So number one, the expression. So you're right. The expression of whatever the enhancer is controlling. So let's say the enhancer is controlling the expression of gene X. Okay, and this enhancer now has transcription factor binding sites for A, B, C, D, E. Gene X's expression is conditional on the presence of then A, B, C, D, E. Okay, so that is kind of one of the better ways of looking at this. You're right. Which of these transcription factors then is kind of causing the nucleosomes to be evicted is not clear. Yeah, and so it could be that one of these transcription factors is. It could be that these transcription factors are not sufficient to evict nucleosomes, and instead you need a pioneering transcription factor that comes before it to clear the path for these transcription factors to bind as well. Okay. Yeah.
So if the genome or the gene is is not bind to the nucleosome, then only one transcription factor is sufficient for the expression of the gene.
Maybe it may not be. Okay. So it could be that it's not sufficient. Yeah.
Okay.
Yeah. But it's going to depend from gene to gene. Okay. Okay. Yeah. So I can see Anindita's question now. So the question is, what are the factors or cues that cause pioneer factors to initiate access to the enhancer sequence, and how do TADs get their cue to alter chromatin topology? And so I think you just asked kind of a fundamental question to what people are trying to address in this system. So I mean, how do pioneer factors initiate access to enhancer sequences? So how are they evicting nucleosomes? That has to be something almost biochemical, I think. This is my bias talking. It's got to be something almost biochemical that will initiate kind of a displacement of the nucleosomes from a particular cluster of sequences. I don't know how that happens, and I'd like to find out. How do TADs get their cue to alter the chromatin topology? Again, that's not entirely clear as well. There are, so for example, there is nuclear actin, there are nuclear myosins as well. There's interactions with the nuclear lamina which seem to play a role in TADs changing their chromatin topology. But if I talk any further, the only thing you'll realize is not how TADs alter their chromatin topology, but how little I know about this. Okay, so ask people who may know better. Someone like Dimple or Anjana may know this in much better detail than I do. Okay, or Bhavna even. Okay. Okay. So like I said, enhancers influence higher order structures to regulate developmental gene expression. We have these particular TADs, and so this concept now takes then groups of enhancers that may be organized in TADs and thinks about evolving new regulatory elements in the context of duplicating TADs, for example. And so, for example, some work that, so you can imagine ways in which particular TAD boundaries, and so TADs, of course, are defined on either end by insulator sequences, or they could be defined by particular housekeeping genes that prevent TADs from migrating into that region. If you lose those insulating sequences, here in this concept, TAD1 and TAD2 could become merged, and so this causes the loss of a particular gene. It could also cause new sequences to arise, which will act as de novo regulatory sequences as well, and so on. Okay, and I don't really want to go into the details of this figure even more, because I want to encourage you to read this paper, okay, or this review. Okay, so the idea of modular enhancers, like I said, is very attractive. This can enable us now to think of a way how different genes can be, how different types of regulation can be evolved. I kind of want to leave you thinking a little bit about the evolution of various networks that control development. And one of the most famous is the Hox complex. And so I'm guessing many of you have investigated or have been lectured about Hox genes during briefly. Okay. And sorry, Shubha, this is also going to be brief as well. Okay. Hox genes, as you probably all know, are a highly conserved cluster of transcription factors, and so they're conserved. They're found in flies, in lamprey, and in mammals. Okay, and many other species as well. They organize so that the most posterior and anterior Hox genes are all in a row along the chromosome, and this kind of organization and regulation seems to have been preserved over almost 1 billion years of evolution. Okay, and so this kind of implies that then the regulatory elements could have been preserved as well, that various ways that Hox genes are regulated may have been preserved as well. Okay, and I will let you investigate this a little bit more. Okay, when we're thinking about this kind of preservation of gene function, one of the classic examples is of PAX6, which is a transcription factor. It will cause a reduction of the eye in humans, mouse, and flies, and so PAX6 seems to be enabling a genetic program that's preserved again over 1 billion years of evolution, and how this is happening is kind of not incredibly clear, to be honest, because when we think about, of course, the photoreceptors of human, mouse, and flies, so the photoreceptors between a fly and a mouse are incredibly different. Okay, so we think of the vertebrate eye, the arthropod eye, and the squid eye, which is also dependent on PAX6, as being very different kind of either compound or kind of this binocular camera eye. And so somehow, even though the eye in insects and in squid and invertebrates has taken on this different developmental route, these different pathways, PAX6 seems to be sitting at the bottom of this developmental program and playing an important role in making the eye. And how that happens is still quite mysterious. Okay, so I think that's all I wanted to talk about. I wanted to talk about the regulation of genes. I wanted to give you a flavor of kind of these slightly evolutionary developmental biology questions as well, which which you can think about and kind of discuss whenever you want. But that's kind of it for today. If you have any questions, please ask.
I have a question.
Yep.
So if you have to just inhibit gene-specific expression of some gene,
Yeah.
I mean tissue-specific expression.
Yeah.
So is this way that we can target these enhancers to achieve that?
Yeah. So there is, you can definitely target enhancers. It's very, it's now very straightforward as well. We can talk about this on Wednesday if you want. I was planning to talk about new techniques to understand developmental biology on Wednesday, but if you want me to be more specific now, I can do.
Then we can do it on Wednesday.
Yeah. So it's essentially just using CRISPR-Cas9 targeting of those enhancers to delete enhancers. Okay. Yeah. Okay. Cool. Okay. Okay. Any other questions? Great. So if there's nothing else, I'll stop there. Hopefully stop. And